Hello, students. So today we are going to talk on chapter 16, the molecular basis of inheritance. We talked earlier about the DNA structure. It was James Watson and Francis Crick who introduced the double helical model of DNA. We know what DNA consists of. It consists of several thousand nucleotide sequences all along the long twisted ladder. It is the code of life, which contains the blueprint of an individual. And this DNA is directing the development of the anatomical, the physiological, and the biochemical, as well as behavioral traits of an individual. I've explained DNA replication process, where DNA replication is the process of making identical copies of itself. And during that process, if any damage occurs during the DNA replication process, the cells can repair their DNA. We know that the role DNA plays in heredity was first discovered studying bacteria and viruses that infect them. The first two scientists, as we know, who put forth this helical model was James Watson and Francis Crick. And now it was a scientist by the name of T.H. Morgan, Thomas Hunt Morgan, who showed that genes are located on chromosomes and that chromosomes are composed of both DNA and protein, which is the genetic material of the cell. So in this chapter, they want to prove that it was the DNA, which is the molecule of inheritance. It is not chromosomes that is the basis of inheritance. So coming back here now, we see that Thomas Hunt Morgan carried out experiments, and then later it was Frederick Griffith in the year 1928 who worked with two strains of a bacterium. One was called the pathogenic strain, pathogenic meaning the one that causes the disease or infection, and one was the non-pathogenic strain, which was the harmless strain. So when he mixed heat kill remains of the pathogenic strain with living cells of the non-pathogenic or the harmless strain, some of the living cells became pathogenic. Some of the living cells caused the infection. So he called this phenomenon transformation, and we are going to be looking at transformation in the lab this Saturday. What is DNA transformation? And in transformation, we've seen that there is a change in both the phenotype, the physical traits, as well as the genotype on account of the foreign DNA that is assimilated. And so when I talk about the DNA transformation here, I want to explain to you, we saw in the previous lab how we used restriction enzymes to cut the DNA. We isolated from bacteria specific sequences, but the restriction enzymes, they cut the sequences of the DNA into smaller fragments. And those small fragments were now referred to as the restriction enzymes. And we use restriction endonucleases, or sometimes we just call them restriction enzymes or REs. And so now we are seeing in DNA transformation, which is the process of taking a foreign DNA and introducing it into a cell. And transformation of the bacteria and the plasmids, which is a circular piece of DNA, is going to be used in biotechnology. And we see that both the bacterial cell will have a starting point, which is called the bacterial origin, which is the point of replication. And then we'll see, and I'll go into details in the lab about an antibiotic resistance gene that was used as a selectable or a specific marker in bacteria. So just to give you a broad perspective of what exactly is DNA transformation. But coming to the experiment that Frederick Griffith carried out on the two strains of bacteria when he took living R cells, which was the non-pathogenic strain which acted as a control, and he injected it into the mouse was healthy. When he took a mixture of heat-killed S cells, the non-pathogenic, and he took S cells, the heat-killed S cells, and the living R cell, injected it into the mouse died. But when he just heat-killed, subjected the S cells, the non-pathogenic strain, the mouse lived. So he said that there was something that was present that caused the non-pathogenic strain to become pathogenic. So later, more scientists came by, Oswald Avery, Macklin McCarty, and Colin MacLeod. I list them by their last names, Avery, MacLeod, and McCarty. They identified that DNA was the transforming substance. So they carried out experiments further to prove that, you know what, it is DNA that is the transforming substance that caused the non-pathogenic strain to become pathogenic. So more evidence for DNA as the genetic material came from studies that were carried out from viruses that infect the bacteria. And what then is a bacteriophage? A bacteriophage, or we simply call it a phage particle, they are viruses that infect a bacteria. A virus is DNA. Sometimes it could be RNA, and it is enclosed by a protective coat. Mostly it is protein, the coat. Now later, it was Hershey and Martha Chase. Alfred Hershey and Martha Chase in 1952, they showed that DNA is the genetic material of this bacteriophage that was known as T2 phage. So they designed an experiment to show that only one of the two components, either the DNA or the protein of the T2, is going to enter an E. coli cell and during the infection and cause the infection. And they concluded that it was the injected DNA of the phage particle that provides the genetic information. So this is what a virus looks like. It's the phage head. It consists of DNA. It has a vibratile tail. And with the help of the tail, it enters into the bacterial cell, thereby inserting either it can insert its DNA or protein into the bacterial cell and thereby cause the infection. So now an experiment was conducted by Hershey and Chase, wherein what Hershey and Chase did was they specifically looked at E. coli, and they had grown it in radioactive sulfur 35S in the phage protein. And here they took radioactive phosphorus 32P in the phage DNA. So they're taking two components and they're trying to prove now which is the molecule of heredity. Is it the protein or is it the DNA? So when Hershey and Chase took both these tags, radioactive sulfur, which was heavy 35S, and radioactive phosphorus, what he found in this experiment, when they infected the E. coli, one sample with the phosphorus labeled phage and the other with the radioactive sulfur labeled phages, when they then put the phage particles in the Erlenmeyer flask or this conical flask. And here we see that the phage particle had, when it infected the bacterial cell, it had the radioactive protein. Whereas this one, so then they blended it, put it in a mixer, and the agitation, when they blended it, the agitation caused in the mixer, it freed, separated out the phage parts from the cell. So you can see here that the phage DNA was separated from the protein in the phage particles. The centrifuge cells now, they gathered and formed a pellet at the bottom, and then you had the supernatant. So when they looked at the liquid, which is the supernatant, it contained the phage protein that was found in the liquid. They measured this radioactivity in the pellet and in the liquid, and they found that in the pellet, they had the DNA, and in the liquid, the supernatant contained the phage protein. The same procedure was subjected. So they took radioactive phosphorus in phage DNA, blended it again, and on centrifugation, they got a pellet. Here, what did they see? That in the pellet now, the pellet contained the radioactive phage DNA was found in the pellet, and what the liquid would have only the phage particles. So this proved that these forces, that in the phosphorus labeled particles, it was DNA, but not the protein. And in the, it was DNA, but in the sulfur, it was protein and not DNA. That's what the pellet contained. So this suggested that the blender removed much of the protein parts of the phage particles, suggesting that protein likely remained adhered to the outside of the cell during the infection. And since this protein remained outside of the cell during the infection, it could not be replicating the genetic material. So now later it was the scientist by the name of Erwin Chargaff. We learned the Chargaff's rule. A forms double bond with thymine, cytosine with guanine, forming a triple bond. And so we know that the nitrogenous bases were adenine, thymine, cytosine, and guanine. And Chargaff put those rules that the base composition of any DNA is going to vary from species to species. But this is important to understand. In any species, the number of adenine and thymine bases is always equal to the number of cytosine and guanine bases. And so we've seen the two strands of the DNA. They run anti-parallel to each other, with one strand beginning at the 5' position and terminating at the 3'. 5' is the phosphate group, 3' is the sugar group. And we see that the outside, when we looked at the DNA structure, the uprights or the skeletons of the ladder consist of alternating sequences of sugars and phosphates, while the rungs, which are the steps of the ladder, consist of the nitrogenous bases, A double bond T or C triple bond G, as we can see out here. Okay. So now later some more scientists came by the name of Wilkins and Rosalind Franklin. So actually it was Rosalind Franklin and Maurice Wilkins who, through X-ray crystallography technique, they showed that DNA molecule looks like this double helix. But they weren't the ones to coin the term double helix. They paved the way for James Watson and Francis Crick. Through X-ray crystallographic images, James Watson and Francis Crick, by looking at Rosalind Franklin and Maurice Wilkins' images, were able now to coin the term that DNA is a double helix. So that's what the pattern showed. And now Watson and Crick thereby built a model in which the backbones run anti-parallel. We see the opposite directions as I just explained here. So now we see, but how did Erwin Chargaff decide that it was one specific purine, adenine, pairs with thymine, a specific pyrimidine forming a double bond, and cytosine, a pyrimidine, pairs with guanine, a purine forming a triple bond? Because now they worked on the model. And so when they put two purines together, and what were the purines? Adenine and guanine. The purines were too wide. They didn't fit within the parallel lines of the double helix. When they took a pyrimidine and a pyrimidine, and what were the two pyrimidines? Thymine and cytosine. They did not fit because it was too narrow. So that allowed them to draw the conclusion that one specific purine, namely adenine, pairs with one specific pyrimidine, namely thymine, forming a double bond. So purine plus pyrimidine's width was consistent with the X-ray data. So now later, Watson and Crick reasoned that the pairing was more specific, and we saw Chargaff's rule. But what does Chargaff's rule state? Even though that one specific purine is pairing with a pyrimidine, in any organism, the amount of adenine is always equal to the amount of thymine. The amount of cytosine is always equal to the amount of guanine. Okay, and we see how the parent molecule is going to unwind in DNA replication process. So I'm going to talk about DNA replication, and I explain to you the process of DNA replication. So these are the two strands of DNA of the double helix. How does DNA replication take place? DNA replication takes place in 3 steps. Each step requires an enzyme. I explained this to you in the lab class, and I'm going to reiterate it again. So each step will naturally require an enzyme. Therefore, you require 3 enzymes. In the first step, the 2 strands of the double helix are going to separate out. These original strands are called the parent strands. Which enzyme is going to help in the uncoiling and separation is DNA helicase. So DNA helicase is going to separate out the 2 strands of the double helix. 5 prime, 3 prime, original parent strand, 3 prime, 5 prime. So these 2 strands now, each strand, each parent molecule now, they separate out with the help of DNA helicase enzymes. Therefore, each molecule is now, each strand is now going to make a partner strand, which is called the daughter strand or the complementary strand. How does it make it? By copying the information from the parent strands. So these 2 are the original parent strands, 5 prime, 3 prime. They separate out. You have 5 prime and you have 3 prime. 5 prime makes a complementary strand, formation of a new strand complementary to the template strand. And so by pairing, base pairing, if you have A here, base pairs with T, C with G, T with A with T, G with C. T with A, G with C, A with T, T with A, C with G. So in step 2, what happens is you have DNA polymerase enzyme, which is going to join these nucleotide sequences correctly on the complementary strand after base pairing with the parent strand. And then finally, in the 3rd step, both these strands, the parent and the complementary, the parent and the complementary strand have to come back together. They coil together at the intersection point called the replication fork, and that is DNA ligase enzyme. So Watson and Crick semi-conservative model. This was called the semi-conservative. 1 old, which is the original parent strand, 1 new, which is the newly synthesized daughter complementary strand. It predicts that when a double helix replicates, each daughter molecule will have 1 old strand derived or conserved from the parent and 1 newly synthesized strand. Other scientists came by and they had put forth three models. One was the semi-conservative, which was proven to be the right one. Then you had competing models, which were the conservative model and the dispersive. In the conservative model, you had the two parent strands, but the two parent strands are going to rejoin. And dispersive is when each strand is a mix of old and new. So experiments by Messelson and Stahl supported the semi-conservative model. And more than a dozen enzymes and other proteins will participate in that replication. So we see in the conservative model, as we have just said, the two parent strands are going to rejoin. So you have the original makes a copy, and these two parent strands will rejoin. That is not correct. We did the semi-conservative where you had old and new, old and new. That was the right model. And dispersive is a mixture of both old and new, old and new. These two were refuted, the conservative and dispersive. So now and this was also proved with the heavy isotope 15 nitrogen, what they did the experiments. So you can just look through this. Now, when you're looking, just to put into perspective, I explained replication very easily, but you have what we call where the DNA strand opens up, that is referred to as the replication bubble. That's the origin of replication where the two DNA strands are going to separate out. So we talked about replication and the replication fork, but however, we talked about DNA helicase, which is unbinding, but you have other enzymes and proteins, one called single strand binding proteins. From the very term, single strand binding proteins is going to bind to, and it is going to stabilize that single stranded DNA. Now, a lot of mistakes can occur during the replication process. So we have a particular enzyme called topoisomerase. What does topoisomerase enzyme do? It relieves the strand. So if this is the replication bubble, and we see at the replication fork, which is this intersection point, the diameter, as I explained on the board when you were in the lab, the diameter between, or the distance between any two replication forks must be consisting of at least 10 minimum nucleotide sequences. Now, if you have a sugar and a phosphate, and the base comes in the next replication fork, that is an error. So it is topoisomerase enzyme which can relieve the strain of the double helix either by breaking it, by swiveling it back, or by rejoining the DNA strands together. So this is a very important enzyme. All these enzymes are important in replication. And so you see, topoisomerase, it can do that. This is the replication fork. This is the point of intersection. And this is the origin of replication, where replication begins. But keep in mind, always, and if DNA is double-stranded, always nucleotide sequences will be added from the 5' 3' region. Whereas RNA is single strand, the sequences will always be added from 3', 5' direction. And we can see here, we saw what DNA polymerase, it was joining the nucleotide sequences. So it requires a primer for which they can add the nucleotides. And that primer is always moving because it's an RNA primer. It is about 3 to 5 nucleotide sequences. It's a short RNA primer. And this primer, I mean, it would be 5 to 10 nucleotides, short one, 5 to 10 nucleotide sequences long, but it would be added from the 3', 5' position. And when you hear the word primase, ASE, primase is going to start cutting. So it can start an RNA chain from scratch and it adds RNA nucleotides one at a time. RNA primase is going to add the nucleotides one letter at a time, one nucleotide sequence at a time. Okay. We talked about DNA polymerase. Now, what's the difference between replication process in prokaryotes like bacteria rather than in eukaryotes, the rate of elongation at which the nucleotide sequences are being added on the DNA strand is about 500 nucleotides per second in bacteria. But in our cells, human cells, it's only 50 per second. So we've seen how DNA polymerase is joining the nucleotide sequences together. So it adds the nucleotides only to the free 3' end because that's a complementary strand. Remember, it joins it on the complementary strand. Therefore, a new DNA strand can elongate only in the 5', 3' position. Now, along one template strand of DNA, the DNA polymerase is going to synthesize what we call a leading strand, continuously moving towards the replication fork. And to elongate the other strand is called the lagging strand. DNA polymerase will act in the opposite direction. So leading is the strand where the leading strand, it is DNA polymerase is synthesizing continuously, moving in. This is important, this moving towards the replication. This is the origin of replication, and DNA polymerase is going to add it from this strand. Now, this would be the lagging strand. The lagging strand is the strand that comes later. See, to elongate the other new strand called the lagging strand, it must work in the direction away. So it's going away from the replication fork. So if this is the leading strand here, the lagging strand is here. This is the replication bubble, the origin of replication. So it's going opposite. Now, what does this lagging strand contain? You see, it's not a continuous strand. This lagging strand has these segments. So the lagging strand will contain these Okazaki fragments, which are joined together by DNA ligase. Remember, DNA ligase is going to anneal it, going to coil it back. So DNA ligase is going to join all these fragments or segments on the lagging strand. So you have the single strand binding proteins which are going to stabilize the DNA strand. We talked about all this. And we see DNA polymerase 3 is going to start to synthesize again the leading strand. So the synthesis takes place during the replication process takes place where a leading strand, lagging strand will be made. And because our DNA is very long, so as it keeps adding more and more nucleotide sequences on the leading and the lagging strand, you have all these other enzymes that are involved in the replication. So, so you have here the lagging strand is in the opposite 3', 5', and the leading strand is always 5', 3'. It is moving. The lagging strand is opposite to the leading strand, and it will always contain these fragments or segments of DNA, which are called Okazaki fragments. And these fragments will be joined together by DNA ligase. Okay. So we talked DNA replication. You can go through all this even in Mastering Biology. I encourage you to look through the video clippings of all these animations. And in a nutshell, you have each and every enzyme and what the function is. I wanted to reiterate, I like to, and I keep saying this even in the chromosomal basis of inheritance, make a little table, name of scientist, experiment conducted, and conclusion. And I hope, so here we have proofreading and repair DNA. How is DNA proofread? How can it be repaired with the help of these repaired enzymes? So DNA polymerase proofreads the newly synthesized strand, which is the complementary strand, replacing any incorrect nucleotide. So instead of adenine pairing with thymine, adenine went and paired with cytosine. And so you have these DNA polymerase is going to proofread it and correct it. Now you have in mismatch repair of DNA, repair enzymes are going to correct any errors that take place. Like I said, instead of adenine pairing with thymine, adenine goes and pairs with cytosine. That's a mismatch repair of DNA that needs to take place. So DNA, now, I've put these notes down in my biotechnology. What are some errors that can occur during replication process? Why do errors occur? Because of exposure to ultraviolet radiation, nicotine in cigarette smoke, any aerosol particles in the environment, any small particulate matter like the aerosols, your hairsprays, your air fresheners, anything, smoke, smog in the environment, all these are called mutagens. Mutagens are chemical substances that cause a mutation to occur, and they're carcinogenic. Carcinogens are substances that cause cancer. And so all these physical or chemical agents are going to cause the DNA to undergo spontaneous changes. Now we have something called nucleotide excision repair. In nucleotide excision is to cut, repair a nuclease. We talked of endonucleases in restriction enzymes that we carried out in DNA isolation. So in nucleotide excision repair, that nuclease enzyme is going to cut out and replace any damaged stretches of the DNA. Now, eukaryotic chromosomal DNA molecules like us, they have nucleotide sequences at their ends, which are called telomeres, that do not prevent the shortening of the DNA molecules. But what do these telomeres do? In fact, they are going to postpone the erosion of the genes near the ends of the DNA molecules. And it has been proposed that the shortening of the telomeres is connecting to aging. So when an addition, these are the chromosomes, these are the telomeres. So if the chromosomes shorten, that shortening of the telomeres is connected to aging, which will result in that individual aging faster. So shortening of the telomeres may also protect the cells from cancerous growth. Why? By limiting the number of cell divisions that can occur. And there is evidence in telomerase activity in cancer cells, which may allow the cancer cells to persist. And therefore, an enzyme called telomerase catalyzes the lengthening of the telomeres in germ cells, which are the somatic cells. I hope you enjoyed this topic, students.